Rainbow papaya. Hihihi.
Hi, Assalamualaikum. We are Group 1 and I am Amira Najha. I'm going to talk about the rainbow papaya, which is a papaya variety that is resistant towards papaya ring spot virus, RSV. So what exactly is a rainbow papaya? Is it a papaya with colorful flesh or a papaya that will grow in the end of the rainbow? Let's get to know about this special papaya. Let me brief you about the rainbow story. The story is originated from one of the islands located in the United States of America named Hawaii. This is the chronology of the rainbow story. In 1950s, the papaya ring spot virus has infected the Oahu Island. To save the papaya industry, the production moved to the Puna area of the Big Island in 1960s. However, the industry has almost destroyed by 40% reduction in 1997. By then, the US government developed new genetically modified papaya variety named Rainbow, which is resistant to PRSP. This is when the rainbow papaya is produced from the crossing of yellow flesh Kapoho Solo variety with the red flesh Sun Up. Now, we will proceed with the main factors of genetic modified papaya development. Okay, first, potivirus PRSP. Potivirus can be easily widespread and also destructive towards papaya. It can be rapidly transmitted by aphid species. Next is due to height severity. PRSV can cause a severe damage in nearly all the major papaya growing areas of the world, including Brazil, Mexico, India, Thailand, Taiwan, Philippines, and the southern region of China, and of course the origin of rainbow papaya, Hawaii. And the ultimate factor of rainbow papaya creation was to rescue the papaya industry, which is Hawaii's fifth largest crop production that have fallen to 40% reduction. These variety were then commercialized in 1998 and have a lot of farmers to cover their loss.
Hello there. Welcome to Newsline. It's 6 June 2020 and I'm Nurul Akila Farhana binti Anwar Sadat. In today's news, I would like to share about rainbow papaya, which is papaya that is resistant to papaya ring spot virus disease or also known as PRSP. The PRSP diagnosis is very important as it exists in different strains. The PRSP might be confirmed by molecular diagnosis such as ELISA, immunocapture real-time PCR, and also GIPA. The transgenic papaya varieties resistant to PRSP against different virus strains must be developed individually for various papaya growing regions. Generally, crops with resistance to viral disease may be developed through genes derived from viral sequence, providing PDR, which is pathogen-derived resistance, genes from various other sources that can interfere with the target virus, and natural resistance genes. We will now proceed with the rainbow papaya development. The transgenic development can be by food protein-mediated resistance, RNA interference-mediated resistance, and also replicase gene-mediated resistance. Now, I would like to explain on food protein-mediated resistance first. Food protein-mediated resistance refers to the resistance of transgenic plants that produce food protein to the virus from which the food protein gene is derived. The food protein transgenic plant can be used to enhance the production of movement proteins and coordinate the formation and size of virus replication complex, thereby limiting the spread of cell-to-cell infection. Fitch and others developed the transgenic papaya containing the food protein gene resistant to papaya ring spot virus disease using the gene transfer system of immature zygotic embryos with the plasmid construction containing the neomycin phosphotransferase 2 gene. Cheng and others also developed the papaya ring spot virus resistant transgenic papaya using the code protein gene of the Taiwanese strain of PRSB constructed with the Thai-binary vector PBGCP through agrobacterium-mediated transformation. And Gonzalez and others also used gene gun technology when transferring the untranslatable code protein gene for the development of papaya ring spot virus resistant papaya variety. This showed the resistance to homologous PRSB isolates from Hawaii, Australia, Taiwan, Mexico, Jamaica, the Bahamas, and also Brazil. We will proceed with RNA interference mediated resistance. The RNA-mediated gene control mechanism has provided a new platform for developing molecular tools for gene function study and crop improvements. This technique is used for generating disease resistance by suppressing a specific gene or genes. PRSB is an RNA virus containing a single open reading peak translates into a large polyprotein that produces the final protein product. RNA-mediated protection would be effective only when the transgene is highly similar to the attacking virus. The helper component proteinase has been shown to be highly effective suppressor of RNA silencing. Luanhan and others report that the transgenic papaya showed resistance to the disease by suppressing post-transcriptional gene silencing. Based on Tenan, he reported that the mechanism of transgenic papaya resistance against papaya ring spot virus are sequence homology dependent and mediated by RNA via post-transcriptional gene silencing. They found that an untranslatable code protein gene was able to confer resistance to the homologous strain of the virus isolate from the papaya ring spot virus by post-transcriptional gene silencing. I will now explain on replicase gene-mediated resistance. The resistance mechanism is protein-based because the resistant phenotype is influenced by mutations affecting the primary structure of the protein encoded by transgene. The replicase genes are different in structure within various genera. The resistance created by the introduction of replicase gene was first demonstrated for the tobacco mosaic virus in Nicotiana tabacum. The replicase gene with mutation have been shown to be able to confer virus resistance. Chen and others report that replicase gene conferred resistance to the papaya ring spot virus in rainbow papaya. These are the list of the transgenic papaya that has been developed by various research groups through gene technology. And that's all for me. Thank you.
So hi again, Assalamualaikum warahmatullahi wabarakatuh, and I'm Ian. So for this part of the video, I would like to share to you about something that might have been wondering in your mind, whether or not this rainbow papaya can reduce the problem. So from the rainbow story that you have already heard at the beginning of the video, the development of the rainbow papaya has recovered the Hawaiian papaya ring spot virus problems and saved the industry, as it is the main importer of fruits to the US and Canada. On a side note, in Thailand, after hearing the successful story of Hawaiian to be able to create the virus-resistant genes, it was disturbed by the anti-GMO activists that forced the Thai government to destroy the program. And currently, Thailand is still battling the virus, as the papaya is the national staple in the traditional dish of somtam. I'm already hungry just hearing about somtam. So in addition to the statement that I have said before, with the amazing discoveries by the Hawaiian, many other countries that are facing similar problems from the virus and diseases are starting to create this rainbow papaya, with the techniques explained in the previous part of this video. So in order to solve the issues, this rainbow papaya is not just being a resistant-free solution to the papaya ring spot virus, but instead it is environmentally safe and has no harmful effects on human health, in addition to being a nutrition-packed food. Thus, we have no worries about the transgenic papaya that could affect the natural ecosystem of the world. This has been approved and researched thoroughly by both the US Environmental Protection Agency, which is also known as EPA, and the USDA Animal and Plant Inspection Services, which is the APHIS office. This rainbow papaya can be compared to the wild type papaya in terms of tree size, fruit size, morphology, and germinability of the seed. So the tree size differs, where the height of the wild type can be very high, whereas the height of the cultivated type can be very short, but at the average height that we want. So this could impact the efficiency to harvest the fruit itself, since the papaya fruit at the top of the tree. So imagine if you have to harvest a very high papaya tree, whereas the fruit is on the top of the tree, it will be very hard for you to harvest, of course. When compared to the short height for the fruit size, the wild type can be very small with a numerous seed and a very thin flesh of the muscle that we consume. When compared to the cultivated type, where you have a moderate sized papaya, the normal size that we see on the market, with an average amount of seed and a very thick flesh, that would make the consumer very happy. And even for the seeds from the wild type, is different to the cultivated type, which differs in size, germination rate, dormancy, and light sensitivity. The wild type seed have a higher longevity, where it can remain dormant when dispersing the seed in the forest due to unfavorable condition of higher trees present, in compared to the cultivated type, where we have a low rate of dormancy to increase the germination rate of the plant. So this papaya has many beneficial impacts that will be explained later on, but the wild type species will still remain protected and conserved, as it is the natural genetic resources of the species, which can be usable for genetic diversity, for breeding and improvement programs. So I think you've heard enough from me. So what about the positive impacts that I've shared to you? Maybe we should like to share the next present.
Hi, Assalamualaikum. My name is Marina binti Muhammad, and today I would like to share about the positive impacts of papaya. Without biotechnology, there is no papaya industry, said Dr. Dennis Gonsalves, the man who developed rainbow papaya. There are many positive impacts of rainbow papaya. The first one is the rainbow papaya saved the papaya industry. As we know, the old variety of papaya are more susceptible to papaya ring spot virus, PRSV. So, by the introduction of new variety of papaya, saved the papaya industry of Hawaii from collapsing because of papaya ring spot virus. It is named rainbow papaya as it gives second chance to Hawaiian farmers to be able to continue growing papaya and allow to produce premium papaya. The second one is the rainbow papaya prevent crop loss. As we know, PRSV reduce more 40% of production of papaya. By planting rainbow papaya variety, it helps farmers to prevent crop loss due to PRSV. The third one is rainbow papaya will increase food supply. The world population is expected to rise 10 billion in 2050. We must boost agriculture demand for protein, fruits, and vegetables. Hence, the GM papaya can improve harvest, which led to increased food supply. It is really important to address, especially in developing countries, to improve food security. Next, the rainbow papaya can easily accessible and affordable. If less amount of papaya produced, the demand will be high and the price of papaya will be increased. So, rainbow papaya can produce higher production of papaya and become more accessible and affordable to consumers. All the consumer, especially underfed nations, can afford to pay more for papaya. The last one, the rainbow papaya provides lots of nutrients. So, by introducing rainbow papaya, it provides consumer carbohydrate, protein, fiber, potassium, and others. Rainbow papaya can alleviate 2 from 3 big macronutrients deficiency that plague undernourished people globally. Okay, that's all from me. Bye! Oh, wait! What are the ethical issues on rainbow papaya?
Hi everybody, my name is Nurul Fadini Saiful Lazim. So today I'm gonna talk about negative impacts and ethical issues of rainbow papaya. So let's dig in. So we're gonna talk about the effects to the environment and to the human health. A recombinant virus has potentially negative effects on the environment, such as increasing pathogenicity, expanding host range, and changing the vector. So far, no recombination has been found in PRSV transgenic papaya in the field. P. Ronwade et al. 2007 reported that there were no distinct differences of microbial community in soil samples collected from transgenic and non-transgenic papaya fields. On the other hand, UniSZA and Penn 2006 reported that PRSV-resistant transgenic plants had a limited effect on the microbial community of the soil. CP expressed by the transgene within a transgenic plant may enter another invading virus infecting the plant via a process of heteroencapsidation. The CP gene can carry determinants for pathogenicity, and the properties of the virus in transgenic plants might be changed. So, vector non-transmissible virus in transgenic plants will be transformed into a transmissible virus through heteroencapsidation, resulting in new viral epidemics. But heteroencapsidation in transgenic papaya expressing virus CP gene has limited significance in practice, and environmental effects will be negligible. Food safety concerns toxins and allergens. Transgenic plants have potential allergenic properties due to the protein encoded by viral sequences. Yeh and Gonzalez, 1994, reported that there were no ill effects after the consumption of PRSV-resistant transgenic papaya. Also, Fermin et al. 2011 observed that transgene-derived PRSV-CP did not pose any risk of food allergies. Benzyl isothiocyanate (BITC) is considered an anti-nutrient which has been identified in extract of Cruciferum, Caricaceae, and Moringaceae. BITC may endanger the fetus in pregnant women and enhance the risk of prostate cancer. The BITC value of transgenic papaya and non-transgenic papaya is similar, and consequently transgenic papaya does not pose any increased threat to human health. Till now, there were no evidence shown that papaya rainbow had negative effect to the environment and human health. Therefore, PRSV-resistant papaya is environmentally safe and suitable for human consumption. So, do you prefer to eat one transgenic papaya? That's all from me. Bye!